Imagine yourself floating in an immense star-dusted void, a silent observer witnessing the birth of worlds. Below you, a young planet glows with primordial heat, its surface a canvas of cooling rock and swirling nascent oceans. It's a scene of profound beginnings, and from this vantage point, the most fundamental question starts to whisper through the stillness. What exactly is this delicate, tenacious phenomenon we call life? How does it distinguish itself from the mere chemistry of the cosmos? This evening we embark on a gentle journey, scaling conceptual levels from 1 to 50, to unravel this profound mystery, starting right at the very edge where non-life meets the first hesitant flicker of being. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. Now, dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. Our exploration begins at level 1, the living question, as we softly explore the core characteristics that we, from our earthbound perspective, associate with being alive. It seems simple enough, doesn't it? A rock is not alive, a tree, a bird, a bacterium. These clearly are. But when you try to pin down a single definitive criterion, the edges begin to soften. We often speak of life possessing metabolism, that intricate dance of chemical reactions where an organism takes in energy and matter from its surroundings, transforming them to build itself, to repair damage, and to power its activities. Think of it as a tiny, self-sustaining chemical factory, constantly processing resources. Then there's reproduction, the ability to create copies of oneself, passing on hereditary information to a new generation. This ensures the continuity of life, a thread woven through time. From the simple division of a single cell to the complex courtship rituals of animals, this drive to perpetuate is fundamental. We also see growth and development, an organized increase in size and complexity, guided by an internal blueprint. A seed doesn't just swell, it unfurls into a structured plant. An embryo doesn't just expand, it differentiates into a complex organism. Living things also exhibit responsiveness, an ability to sense and react to changes in their environment. A sunflower turns towards the light, an amoeba recoils from a noxious chemical. This interaction with the surroundings is crucial for survival and for finding resources. And perhaps most profoundly, life on Earth shows evolution by natural selection, the capacity for populations to change over generations, adapting to their environment as individuals with advantageous traits are more likely to survive and reproduce. Yet as we move to level 2, the blurry line with non-life, you encounter entities that challenge these neat boxes. Consider a virus. It has genetic material, it reproduces, though only by hijacking a host cell's machinery, and it certainly evolves. But it has no metabolism of its own. Is it truly alive? Scientists still debate this, highlighting that our definitions born from studying Earth life might not be universal. A crystal can grow in a highly ordered way, adding new layers, but it doesn't metabolize or evolve in a biological sense. Fire, as we've noted before, consumes fuel, grows, and can even reproduce by spreading sparks, but it lacks the complex, information-based heredity of life. This blurring becomes even more pronounced when we consider the very earliest steps towards life, a period where complex chemical systems might have exhibited some, but not all, of these characteristics. Perhaps there were self-replicating molecules that weren't yet enclosed in a cell, or simple vesicles that could grow and divide but lacked a sophisticated genetic system. These proto-life entities would occupy a fascinating grey area. The transition from non-life to life was likely not an instantaneous switch, but a gradual emergence of increasing complexity and integration of these lifelike properties. Philosophers and scientists continue to grapple with whether a single, universally applicable definition of life is even possible, especially when we consider the potential for truly alien life based on different chemistries. As we contemplate this blurry boundary, we must also consider level 3. Early energy sources for no life, however simple, can exist without a source of energy to drive its processes and maintain its organization against the universe's natural tendency towards disorder or entropy. On the primordial Earth, energy was abundant and came in many forms. The young sun, even if fainter overall, bathed the planet in intense ultraviolet radiation, unshielded by an ozone layer that had yet to form. This UV light could break chemical bonds and provide the activation energy for new reactions to occur. Lightning in the turbulent early atmosphere would have been a spectacular and frequent phenomenon, discharging immense electrical energy into the gases and surface waters, again driving chemical synthesis. Volcanic activity, far more widespread than today, provided geothermal heat. Hot springs and deep-sea hydrothermal vents released geothermally heated, mineral-rich fluids, creating localized environments with strong chemical and thermal gradients. Natural batteries, in a sense, that could power reactions. Even the shock waves from meteorite impacts, a common occurrence in the early solar system, could have briefly created conditions of extreme temperature and pressure, capable of forging new molecules. Understanding these available energy sources is crucial because they represent the power supply for the very first chemical experiments that could have led to life. Just as a cook needs heat to transform raw ingredients, these natural energy sources were the flames under Earth's primordial cauldron, enabling simple inorganic molecules to combine and react, forming more complex structures. The type and intensity of energy available would have influenced which chemical pathways were favored and where the most promising experiments might have taken place. Scientists still explore the relative contributions of these different energy sources. Was it the sun's rays in shallow ponds, the electrical storms in the atmosphere, or the chemical energy from deep-sea vents that played the most critical role in jump-starting the chemistry of life? The answer is likely a combination, with different environments and energy forms contributing to the rich chemical tapestry of early Earth. So as we begin our ascent through these levels of understanding, we start with this fundamental realization. Life, while seemingly distinct, emerges from a continuum of complexity, powered by the raw energies of a young and dynamic planet. The definition is elusive, the boundary with non-life permeable, and the available energy diverse, a perfect setup for nature's grandest experiment. Having acknowledged the energetic landscape of the young Earth and the elusive nature of a strict definition for life, we now advance our understanding, delving into Level 4, Primordial Earth's Atmosphere. Imagine yourself trying to breathe on this nascent world billions of years ago. The air would be utterly alien, a stark contrast to the oxygen-rich blanket we depend on today. Free oxygen, O2, the kind we breathe, was virtually absent. Instead, the early atmosphere was likely what scientists call a reducing or perhaps a neutral environment. This terminology refers to its chemical properties. A reducing atmosphere is rich in molecules that can donate electrons, like hydrogen, methane, and ammonia, while a neutral one might be dominated by carbon dioxide and nitrogen. Volcanoes, far more numerous and active than today, were the primary architects of this early sky, constantly spewing forth a cocktail of gases from the planet's interior. Water vapor, H2O, would have been a major component, billowing from these volcanic vents and contributing to a steamy, humid environment once the planet cooled sufficiently for liquid water to exist. Carbon dioxide, CO2, would also have been abundant, released from magmatic processes. Nitrogen, N2, a relatively stable and unreactive gas, likely formed a significant portion, much as it does in our modern atmosphere, where it makes up about 78%. The presence and concentration of other gases like methane, CH4, ammonia, NH3, hydrogen sulfide, H2S, and free hydrogen, H2, are subjects of ongoing scientific debate and are crucial for models of prebiotic chemistry. Early theories, like those underpinning the Miller-Urey experiment, which we will touch upon soon, favored an atmosphere rich in methane and ammonia, as these compounds readily form amino acids and other organic molecules when energized. However, more recent geological and atmospheric models suggest that the early atmosphere might have been less strongly reducing, perhaps dominated by CO2 and N2, with smaller amounts of methane and ammonia. This doesn't preclude the formation of organic molecules, but it might mean that different pathways or locations, like hydrothermal vents, were more important for their synthesis. The exact composition of this early atmosphere is a critical variable because it dictates the starting ingredients available for life's first chemical steps and influences surface temperatures through the greenhouse effect. As the planet continued to cool from its fiery formation, this abundant atmospheric water vapor began to condense, leading us to level 5, the first oceans. Imagine torrential, planet-scouring rains lasting for perhaps thousands or even millions of years, gradually filling the vast, impact-scarred basins of the early Earth. These primordial oceans would have been very different from the salty seas we know today. Initially, they might have been freshwater, or perhaps slightly acidic, due to high levels of dissolved CO2 forming carbonic acid. Their temperature would have been warm, possibly even hot in places, heated by ongoing volcanic activity on the seafloor and the residual heat from Earth's accretion. These early oceans became a vast chemical repository. They dissolved gases from the atmosphere, leached minerals from the primitive crustal rocks they lapped against, and became the collecting basin for materials delivered by comets and meteorites. This created a complex aqueous solution, a kind of global broth where countless chemical reactions could occur. The interface between the water and the atmosphere, and between the water and the seafloor, would have been particularly dynamic regions for chemical activity. The presence of liquid water is considered absolutely critical for life as we know it, acting as a superb solvent, a medium for transporting molecules, and a direct participant in many biochemical reactions. The emergence of these first oceans was a pivotal moment, transforming Earth into a water world and providing the vast liquid arena necessary for the chemistry of life to unfold. Within this atmospheric and oceanic crucible, we find level 6, key inorganic molecules present. These are the simpler, non-carbon backbone molecules that served as the fundamental starting materials for more complex organic structures. We've already mentioned water, H2O, carbon dioxide, CO2, nitrogen, N2, methane, CH4, and ammonia, NH3. Hydrogen sulfide, H2S, released from volcanoes, would have been a source of sulfur. Phosphate, PO4.3, crucial for the backbone of DNA and RNA and for cellular energy transfer, ATP, would have been leached from rocks, though its availability in a soluble, reactive form is sometimes considered a potential bottleneck in early prebiotic chemistry. Simple metal ions, dissolved from rocks, would also have been present and could have played catalytic roles in early chemical reactions. Think of this stage like setting up a vast planetary-scale laboratory. You have your primary solvents, the oceans, your gaseous reactants, the atmosphere, and a range of dissolved inorganic salts and minerals. The energy sources we discussed in the previous section, UV radiation, lightning, geothermal heat, are now acting upon this specific chemical inventory. The composition of the atmosphere determines which gases are directly exposed to atmospheric energy sources. The composition of the oceans determines what's available in solution and what kinds of reactions can occur at mineral-water interfaces on the seafloor or in shoreline environments. Scientists still debate the precise concentrations and relative importance of these various inorganic molecules. For instance, was the early ocean rich in dissolved iron, as some evidence suggests from ancient rock formations? This could have influenced the types of chemical reactions possible and the availability of certain catalysts. The pH of the early ocean, whether it was acidic, neutral, or slightly alkaline, is also a key parameter that would have affected chemical stability and reactivity. The core concept here is that before you can build complex organic structures like proteins and nucleic acids, you need a readily available supply of their simpler inorganic precursors and the elements they contain: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. The early Earth, through volcanic outgassing, the condensation of its oceans, and the interaction between rock, water, and air, provided this essential starting toolkit. It was a world primed for chemical evolution, a crucible where these simple inorganic molecules, under the influence of abundant energy, were about to embark on a journey towards increasing complexity, leading to the first organic building blocks of life, a process we will explore as we ascend to the next levels. The primordial soup wasn't just water. It was a dynamic chemical system, a complex interplay between the atmosphere, the oceans, and the solid Earth, all simmering under a young, energetic sun. Having established the dynamic environment of early Earth, with its unique atmosphere, nascent oceans, and abundant inorganic molecules, we now ascend to a crucial stage in unraveling life's mystery: the emergence of the first organic monomers. This is where we move from the realm of simple inorganic chemistry to the more complex carbon-based molecules that form the very foundation of life as we know it. We're looking at Level 7, how simple building blocks like amino acids formed, followed by Level 8, the genesis of nucleotides, and finally, Level 9, considering other key precursors like sugars and lipids. Let's start with amino acids, the molecular units that link together to form proteins. Proteins are the workhorses of modern cells, performing a vast array of functions. They act as enzymes to catalyze biochemical reactions, provide structural support, transport molecules, and much more. There are 20 common amino acids used by life on Earth, each with a central carbon atom bonded to an amino group, NH2, a carboxyl group, COH, a hydrogen atom, and a unique side chain, the R group, that gives each amino acid its specific properties. The question is, how could these relatively complex molecules have formed spontaneously on the early Earth from the simpler inorganic precursors we've discussed, such as methane, CH4, ammonia, NH3, water, H2O, and carbon dioxide, CO2? This is where the famous Miller-Urey experiment, conducted in 1952 by Stanley Miller under the guidance of Harold Urey, provided a groundbreaking insight. As we touched upon, they created a closed system containing a mixture of gases then thought to represent Earth's early reducing atmosphere, methane, ammonia, hydrogen, and water vapor. They then subjected this mixture to electrical sparks, simulating lightning. After just a week of continuous operation, the water in the apparatus, which had turned a murky reddish-brown, was found to contain a variety of organic compounds, including several different amino acids. This experiment was a landmark because it demonstrated for the first time that the essential building blocks of proteins could be synthesized abiotically without the involvement of living organisms under plausible early Earth conditions. It showed that the leap from simple inorganic gases to the molecules of life was not an insurmountable chemical hurdle. While the exact composition of the early atmosphere Miller and Urey used is now debated by some scientists who favor a more neutral atmosphere, richer in CO2 and N2, subsequent experiments using different gas mixtures and energy sources, like UV radiation or shock waves simulating meteorite impacts, have also successfully produced amino acids and other organic molecules. For instance, even with more CO2-rich atmospheres, the presence of some methane or the addition of compounds like hydrogen sulfide can significantly enhance amino acid yields. Furthermore, some theories suggest that amino acids could have formed in specific localized environments, such as near volcanic plumes, rich in reducing gases, or even in deep-sea hydrothermal vents, where chemical gradients and mineral catalysts are abundant. The key principle remains: energy acting upon a mixture of simple inorganic molecules can lead to the spontaneous formation of amino acids. Now let's consider Level 8, the genesis of nucleotides. Nucleotides are even more complex monomers than amino acids, and they are absolutely essential as the building blocks of nucleic acids, DNA and RNA, which carry genetic information. Each nucleotide consists of 3 parts: a phosphate group, a 5-carbon sugar, deoxyribose in DNA, ribose in RNA, and a nitrogenous base, adenine, guanine, cytosine, thymine in DNA, adenine, guanine, cytosine, uracil in RNA. The abiotic synthesis of complete nucleotides, especially the sugar component, ribose, and its correct linkage to the base and phosphate, has proven to be a more challenging chemical puzzle than that of amino acids. Ribose, the sugar in RNA, is notoriously unstable and can degrade easily. Moreover, forming it prebiotically often leads to a complex mixture of many different sugars, making it difficult to select for just ribose. However, scientists have made significant progress in recent years. For example, some pathways propose that the sugar and the nitrogenous base might have formed separately and then combined, while other, more recent and compelling scenarios suggest that components of the sugar and the base could have formed from common precursors and assembled in a more integrated fashion. Researchers like John Sutherland and his team have demonstrated plausible chemical pathways using simple starting materials like cyanamide, cyanoacetylene, and glycolaldehyde, all thought to be available on early Earth, that can lead to the formation of ribonucleotides under conditions involving UV light and wet-dry cycles, perhaps in shallow ponds. These pathways often bypass the separate formation of free ribose, instead building up the nucleotide structure in a more stepwise, interconnected manner. The exact mechanisms are still an active area of research, but these studies show that the formation of nucleotides from simple prebiotic precursors is not chemically impossible. Moving to level 9. Considering other key precursors, we also need to account for the origin of simple sugars, beyond ribose, which are used for energy and structure, and lipids, which form cell membranes. Simple sugars like glucose could potentially form from formaldehyde, CH2O, through a reaction called the Formose reaction, though this reaction tends to produce a complex tarry mixture of many different sugars. How specific sugars were selected and utilized by early life is still a question. Lipids, or rather their simpler precursors like fatty acids, are crucial for forming the boundaries of the first cells. Fatty acids are long hydrocarbon chains with a carboxyl group at one end. These molecules are amphiphilic, meaning one end is hydrophilic, water-loving, and the other is hydrophobic, water-fearing. This property allows them to spontaneously self-assemble into vesicles, micelles or bilayers, in water, forming primitive membranes. Fatty acids can be synthesized through reactions like the Fischer-Tropsch process, which involves hydrogen and carbon monoxide reacting over mineral catalysts at moderate temperatures, conditions that might have existed in certain geothermal environments or been associated with meteorite impacts. They have also been found in meteorites, suggesting an extraterrestrial source as well. Think of this stage as a cosmic molecular construction yard. The inorganic molecules are the raw bulk materials like sand, cement, and water. The energy sources are the power tools, and the organic monomers, amino acids, nucleotides, fatty acids, simple sugars, are the first prefabricated components like bricks, electrical wiring, and plumbing pipes. These components are now ready to be assembled into more complex structures. It's important to remember that the early Earth wouldn't have produced these monomers in a clean, purified state. It would have been a complex, messy mixture, a primordial soup, or perhaps more accurately, a primordial slush, or primordial pizza, depending on whether these reactions occurred in solution, on surfaces, or in fluctuating environments. Scientists still debate the precise locations where these monomers formed and accumulated in sufficient concentrations to allow for the next step: polymerization into larger molecules like proteins and RNA. Was it in shallow evaporating ponds, within the porous structures of hydrothermal vents, on the surfaces of clay minerals, or perhaps in ice eutectic phases, where freezing water can concentrate solutes? Most likely, a combination of these environments contributed to the rich organic inventory of the prebiotic Earth. The mystery is not so much whether these building blocks could form, but which specific pathways and environments were most productive and led to the particular set of molecules utilized by life. Having established how the fundamental building blocks of life, amino acids, nucleotides, lipids, and sugars, could have emerged from simpler precursors on the early Earth, we now ascend to a fascinating and widely influential concept regarding the next crucial steps towards life, the RNA world hypothesis. This theory addresses the chicken and egg problem of early life. In modern cells, DNA stores the genetic information, and proteins, enzymes, carry out most of the catalytic work, including the replication of DNA and the synthesis of proteins themselves. So which came first? If DNA needs proteins to replicate, and proteins need DNA to be encoded, how could such a system have gotten started? The RNA world hypothesis proposes a compelling solution: that an earlier form of life, or proto-life, was based primarily on RNA, with RNA molecules serving both as the genetic material and as the primary catalytic molecules. Let's break this down, starting with level 10, RNA's potential to store information. Like DNA, RNA is a nucleic acid made up of a chain of nucleotides. Each RNA nucleotide contains a ribose sugar, a phosphate group, and one of 4 nitrogenous bases: adenine, A, guanine, G, cytosine, C, and uracil, U, which replaces thymine found in DNA. The sequence of these bases along an RNA strand can encode genetic information, just as the sequence of bases in DNA does. While RNA is typically single-stranded, it can fold back on itself to form complex 3-dimensional structures, including double helical regions where A pairs with U and G pairs with C, similar to DNA base pairing. This ability to carry sequence information and adopt specific structures is fundamental for a molecule to act as a genetic material. In an RNA world, these RNA sequences would have contained the blueprints for early life forms, guiding their replication and perhaps the synthesis of other useful RNA molecules. Next, we consider level 11, RNA's capacity to catalyze reactions. This is perhaps the most revolutionary aspect of the RNA world theory. For a long time, it was believed that only proteins with their complex folded structures and diverse amino acid side chains could act as biological catalysts, enzymes. However, in the 1980s, Sidney Altman and Thomas Zech independently discovered that some RNA molecules can also possess enzymatic activity. These catalytic RNA molecules are called ribozymes. This discovery, for which they received the Nobel Prize in Chemistry in 1989, was a game changer. It demonstrated that RNA was not just a passive carrier of information, but could also actively participate in and facilitate chemical reactions. Ribozymes can catalyze a variety of reactions, including the cutting and splicing of RNA strands, the formation of peptide bonds, the links between amino acids in proteins, and even the replication of short RNA templates. They achieve this by folding into specific 3-dimensional shapes that create active sites, much like protein enzymes do. These active sites can bind to other molecules, substrates, and lower the activation energy required for a reaction to occur. The existence of ribozymes provided strong evidence that RNA alone could have managed the essential catalytic functions of early life, without needing a pre-existing suite of protein enzymes. This brings us to level 12, the dominance of an RNA world. The hypothesis posits that for a significant period in the early history of life, RNA molecules were the central players. In this scenario, primitive cells or protocells would have contained a collection of RNA molecules. Some RNAs would store the genetic heritage. Other RNAs, ribozymes, would catalyze the replication of these genetic RNAs, perhaps synthesize other necessary RNA components, facilitate the formation or maintenance of the protocell's boundary, or even carry out simple metabolic reactions. It would have been a world where RNA did it all, heredity and catalysis. What evidence supports this ancient RNA dominion beyond the existence of ribozymes? The ribosome. As we've noted, the ribosome, the cellular machinery responsible for synthesizing proteins from mRNA templates, is itself a giant ribozyme. The catalytic core of the ribosome, where peptide bonds are formed, is composed of ribosomal RNA, not protein. This is considered a strong molecular fossil, hinting that protein synthesis itself originally evolved in an RNA-based system. RNA cofactors. Many essential enzyme cofactors, small helper molecules that enzymes need to function, in modern metabolism are RNA nucleotides or contain RNA-like structures. ATP, adenosine triphosphate, the universal energy currency, is a ribonucleotide. Others like NADH, FADH2, and coenzyme A also have ribonucleotide components. This suggests that RNA was deeply involved in the earliest metabolic pathways. Simpler synthesis. Some researchers argue that the building blocks of RNA, ribonucleotides, might have been somewhat easier to synthesize under plausible prebiotic conditions than the deoxyribonucleotides needed for DNA, though the abiotic synthesis of ribose itself remains a complex challenge, as we discussed. Versatility. RNA's ability to be both an information carrier and a catalyst makes it a uniquely versatile candidate for early life, elegantly solving the DNA-protein chicken and egg problem. If an RNA world existed, how did life transition to the DNA-protein-based system we see today? This would have been a gradual evolutionary process driven by the advantages of specialization. DNA is chemically more stable than RNA due to the deoxyribose sugar and its typical double-stranded nature, making it a better long-term repository for genetic information, especially as genomes grew larger and more complex. Proteins, with their 20 different amino acid building blocks, can form a much wider variety of three-dimensional structures and perform catalytic functions with greater efficiency and specificity than most ribozymes, which are limited to 4 bases. Perhaps early RNA-based systems began to synthesize simple peptides, and some of these peptides by chance enhanced the stability or catalytic activity of the RNA molecules. This would create a selective advantage. The evolution of an enzyme, possibly an early protein or even a ribozyme, capable of reverse transcription, making a DNA copy from an RNA template, would have been a key step, allowing genetic information to be archived in the more stable DNA format. Gradually, proteins would have taken over most catalytic roles due to their superior efficiency, and DNA would have become the primary genetic material, with RNA settling into its modern intermediary roles, mRNA, tRNA, rRNA, and regulatory functions. Think of it like the evolution of a small one-person workshop. Initially, the single craftsperson, RNA, does everything: designs the product, stores information, builds it, catalysis, and manages the shop. As the business grows, it becomes more efficient to hire specialists. A dedicated designer, DNA, to create and store the blueprints with greater care, and skilled workers, proteins, each specializing in a particular aspect of construction, leading to higher quality and output. The original craftsperson, RNA, might then take on important roles coordinating between the designers and the workers. The RNA world hypothesis, while not without its challenges and unanswered questions, such as the robust prebiotic synthesis and stability of RNA and how the first self-replicating RNA emerged, remains a highly compelling and influential framework for understanding a crucial phase in the origin of life. It provides a plausible bridge from a world of simple organic molecules to the complex interconnected systems of DNA, RNA, and proteins that characterize all known life today. It suggests a period of molecular ingenuity where a single type of molecule bravely shouldered the immense task of kick-starting biology. Having explored the tantalizing possibility of an RNA world, where life's early functions were managed by versatile RNA molecules, we now turn our attention to the specific physical environments on primordial Earth where such molecular evolution might have taken place. While sunlit shallow pools and atmospheric reactions have their proponents, another set of compelling candidates for life's nurseries lies in regions of intense geothermal activity. We journey now to level 13, hydrothermal vents, deep on the ocean floor, and also consider level 15 volcanic pools on land, examining their unique chemistry at level 14. Let's first descend into the abyssal depths of the early oceans. Here, far from the reach of sunlight, you encounter hydrothermal vents, fissures in the planet's crust where seawater, having percolated deep within, is heated by underlying magma and then erupts back into the cold ocean. This superheated water, reacting with the surrounding volcanic rocks, becomes laden with dissolved minerals and reduced gases. Black smokers, one type of vent, can spew fluids at temperatures exceeding 400 degrees, 750 degrees, rich in iron and sulfur compounds that precipitate out as dark chimney-like structures. These environments are extreme, yet they offer a continuous supply of chemical energy and raw materials from Earth's interior, shielded from harsh surface UV radiation and frequent meteorite impacts. The discovery of thriving ecosystems around modern hydrothermal vents, entirely dependent on chemosynthesis, deriving energy from chemical reactions rather than sunlight, revolutionized our thinking about habitability. If complex life can flourish there today, perhaps life first originated in such settings. At level 14, exploring their unique chemistry, we find that the porous mineral structures of vent chimneys, with their myriad micro-compartments, could have acted as natural chemical reactors. These compartments can concentrate precursor molecules, preventing their dispersal. The surfaces of minerals like iron sulfides, pyrite, clays, and other metal sulfides possess catalytic properties, potentially facilitating the polymerization of amino acids into peptides or nucleotides into RNA-like strands. A particularly interesting type of vent for origin of life scenarios is the alkaline hydrothermal vent, exemplified by the Lost City hydrothermal field. These vents are typically cooler, 40–90 degrees, than black smokers and release fluids that are alkaline and rich in hydrogen gas, H2, methane, CH4, and simple organic molecules produced through a geological process called serpentinization, the reaction of seawater with mantle rocks. The porous carbonate chimneys formed at these vents provide natural gradients in temperature, pH, and chemical concentrations between the vent fluid and the surrounding seawater. These gradients could have driven the formation of simple organic molecules and even the emergence of primitive metabolic cycles. Some theories, like Michael Russell's, propose that these mineral compartments acted as the first cells, with inorganic walls across which proton gradients, differences in H+ ion concentration, could have been established, providing an early form of energy currency before the evolution of ATP. The continuous flow of reactants and energy, combined with catalytic mineral surfaces and protective niches, makes these alkaline vents a highly plausible environment for the sustained chemical evolution leading to life. Now let's ascend to the surface of the early Earth and consider level 15, volcanic pools as alternative, or perhaps complementary, nurseries. Young Earth was a geologically hyperactive planet, with numerous volcanoes dotting its landscape. Around these volcanoes, geothermal heat would have created hot springs and shallow pools of water. These pools would have been rich in dissolved minerals leached from volcanic rocks and gases from volcanic emissions. What advantages might these volcanic surface pools offer? Concentration through evaporation. Shallow pools are subject to cycles of evaporation and rehydration. As water evaporates, dissolved molecules become more concentrated, increasing the likelihood of them reacting and polymerizing. This is a significant advantage for forming long-chain molecules like peptides or nucleic acids, as the removal of water, dehydration, is often required for monomers to link together. Access to atmospheric gases and solar energy. Surface pools would be directly exposed to the early atmosphere and to solar radiation, including UV light. While high levels of UV can be destructive, moderate levels could have provided the energy needed to drive certain prebiotic chemical reactions. Lightning strikes in the vicinity could also have contributed. Mineral catalysts. Volcanic rocks and clays lining these pools could have provided catalytic surfaces similar to those in hydrothermal vents, promoting the synthesis and polymerization of organic molecules. Wet-dry cycles. The fluctuating conditions of wetting and drying could have been crucial. For instance, some experiments suggest that the formation of peptide bonds or the linkage of nucleotides can be facilitated on mineral surfaces during drying phases, with subsequent rehydration, allowing the newly formed polymers to dissolve and potentially interact further. It's been proposed that such cycles could also help encapsulate molecules within lipid vesicles. Imagine these volcanic pools as dynamic, miniature laboratories. They are constantly being fed new chemicals from volcanic inputs and atmospheric deposition. They are being energized by sunlight and geothermal heat. They are undergoing cycles of concentration and dilution. This fluctuating, energy-rich environment could have been highly conducive to the complex chemical experimentation needed for abiogenesis. Both deep-sea hydrothermal vents and surface volcanic pools offer compelling features as potential cradles for life. Scientists still actively debate the relative merits and likelihood of each. Vents offer protection from harsh surface conditions, UV impacts, and a continuous geochemically driven energy supply. However, concentrating precursor molecules and achieving polymerization in a constantly flowing, open deep-sea system can be challenging. Volcanic pools offer concentration mechanisms, evaporation, and access to solar energy, but they are more exposed to destructive surface conditions. The precise composition of the early atmosphere also heavily influences the types of reactions that would be favored in surface environments. It's also plausible that different stages of life's origin occurred in different environments. Perhaps simple organic monomers were formed in the atmosphere or delivered by meteorites, and then became concentrated and polymerized in volcanic pools. Or perhaps these building blocks were transported to deep-sea vents, where they were further organized within mineral compartments and powered by geothermal chemistry. Some theories even propose a link, suggesting that meteorite impacts could have created temporary hydrothermal systems on the surface. The core concept here is that specific geological settings provided the necessary combination of ingredients, energy, catalysis, and compartmentalization, or concentration, for prebiotic chemistry to progress towards life. Whether it was the dark, steady warmth of a deep-sea vent, with its mineral-rich fluids and protective chimneys, or the fluctuating, sun-drenched and geothermally heated environment of a volcanic pool, these were places where the primordial soup was not just a dilute ocean, but a concentrated reactive brew. Think of them as nature's own test tubes and beakers, where the planet itself was conducting the experiments that would ultimately lead to the first living cells. The where of life's origin is as crucial a question as the how, and both these geothermally active environments remain prime candidates in our quest to understand life's first home. As we continue our ascent through the levels of understanding life's origin, having explored potential terrestrial nurseries like hydrothermal vents and volcanic pools, we now broaden our perspective to consider a crucial external influence: the delivery of essential materials from outer space. It's increasingly recognized that the early Earth was not an isolated chemical system. Instead, it was constantly being showered with material from the cosmos, a delivery service that may have played a vital role in providing the building blocks for life, or even, some speculate, life itself. This brings us to level 16, meteorites carrying organics, level 17, cometary contributions, and the overarching theory at level 18, the broader panspermia theory. Let's first examine level 16, meteorites carrying organics. The early solar system, as we've noted, was a much more chaotic place than it is today, a veritable shooting gallery of asteroids, comets, and planetary debris. Earth, in its infancy, especially during the late heavy bombardment, roughly 4.1 to 3.8 billion years ago, experienced a far higher rate of impacts from these objects. A particular class of meteorites, known as carbonaceous chondrites, has proven to be a treasure trove of information about the chemistry of the early solar system. These meteorites are essentially pristine samples, remnants from the solar system's formation, that have remained largely unchanged for over 4.5 billion years. When analyzed in laboratories, they are found to be surprisingly rich in organic compounds, molecules containing carbon, often bonded with hydrogen, oxygen, and nitrogen. Crucially, these organic molecules are generally understood to have formed abiotically, without biological processes, in space, either in the interstellar molecular clouds from which our solar system condensed, within the protoplanetary disk itself, or through chemical reactions on their asteroid parent bodies, driven by processes like UV radiation, cosmic rays, or aqueous alteration. The Murchison meteorite, which fell in Australia in 1969, is a famous example. It contains a diverse array of organic molecules, including over 70 different amino acids, many of which are not found in terrestrial life, indicating their extraterrestrial origin. Nuclear bases, the building blocks of DNA and RNA, such as adenine, guanine, and uracil. Sugars and sugar-like molecules, fatty acids, which can form membranes, and polycyclic aromatic hydrocarbons, PAHs. The discovery of such a rich organic inventory within these ancient celestial objects provides compelling evidence that the fundamental building blocks of life are not unique to Earth, but are synthesized naturally throughout the cosmos and can be delivered to planetary surfaces. This influx of meteoritic material could have significantly enriched Earth's prebiotic chemical soup, providing a ready-made toolkit for the subsequent steps towards life. Next, we turn to level 17, cometary contributions. Comets, often described as dirty snowballs or icy dirt balls, are conglomerates of ice, water ice, carbon dioxide ice, methane ice, ammonia ice, dust, and embedded organic molecules. They originate from the colder outer regions of the solar system, primarily the Kuiper belt beyond Neptune and the Oort cloud even farther out. Like meteorites, comets are also considered to be relatively pristine remnants from the solar system's formation. As comets approach the Sun, they heat up and their ices sublimate, turn directly into gas, releasing dust and organic compounds, forming the characteristic coma atmosphere and tail. Missions like the European Space Agency's Rosetta mission to Comet 67P Churyumov-Gerasimenko have provided invaluable close-up data on cometary composition. Rosetta's instruments detected a wide array of organic molecules, including amino acids like glycine, various hydrocarbons, alcohols, and even complex carbon-rich macromolecules. Comets are also thought to have delivered a significant portion of Earth's water, especially during the early bombardment phase. If comets brought water, they undoubtedly also brought their embedded organic cargo. The impact of comets, while potentially destructive if very large, could have dispersed these vital ingredients across the young planet's surface. Some theories even suggest that the energy of cometary impacts could have driven further chemical synthesis, transforming simpler delivered organics into more complex ones. This brings us to level 18, the broader panspermia theory. Panspermia, in its most general sense, is the hypothesis that life, or its essential components, is distributed throughout the universe and can be transported from one celestial body to another. We can distinguish a few forms: molecular panspermia, or soft panspermia. This is the idea we've largely been discussing, that the building blocks of life, amino acids, nucleobases, etc., are delivered to planets from space via meteorites, comets, and interplanetary dust. This form of panspermia is widely accepted as having occurred on early Earth and likely contributed significantly to its prebiotic chemical inventory. Lithopanspermia. This is the more radical hypothesis that actual viable microorganisms, perhaps hardy bacterial spores or extremophiles, could be transported through space, embedded within rocks ejected from a planet's surface by a major impact. These life-bearing rocks could then travel between planets within a solar system, or even potentially between star systems, interstellar panspermia, and seed life on other habitable worlds. The challenges for lithopanspermia are considerable. Microbes would need to survive the initial ejection from their home planet, the harsh conditions of space, vacuum, radiation, temperature extremes, for potentially millions of years, and then the fiery atmospheric entry and impact on a new planet. However, we know that some terrestrial microbes are incredibly resilient. Experiments have shown that some bacteria can survive simulated space conditions, especially if shielded by rock. We also have direct evidence of rocks being exchanged between planets within our solar system. For example, numerous meteorites found on Earth have been definitively identified as Martian in origin. While no viable Martian microbes have been found in them, it proves the transport mechanism is possible. If lithopanspermia were to occur, it wouldn't solve the ultimate question of abiogenesis, how life first arose from non-life, but it would mean that life, once started, could be much more widespread and interconnected than if it had to arise independently on every habitable planet. Earth life might have originated on Mars and been transported here, or vice versa, or perhaps it came from even farther afield. Think of the early Earth as a newly tilled garden. Earth-based processes, atmospheric synthesis, reactions in volcanic pools or vents, were like the gardener planting some seeds they had on hand. The cosmic delivery service, meteorites and comets, was like a constant wind blowing in additional seeds and fertilizer from all over the neighborhood, and perhaps even from distant lands. This cosmic seeding could have provided a richer and more diverse starting mix of ingredients than Earth could have produced on its own, potentially accelerating the path to life or providing specific molecules that were otherwise rare. The exact balance between endogenously produced organics, made on Earth, and exogenously delivered organics from space is still a topic of active research. However, the evidence is overwhelming that extraterrestrial materials played a significant role. It connects our planet's biology directly to the chemistry of the wider cosmos, reminding us that the ingredients for life are not a terrestrial monopoly, but are part of the universe's common heritage. This cosmic connection makes the possibility of life elsewhere seem all the more plausible, as the same building blocks would have been delivered to countless other young planets throughout the galaxy. Having explored the potential sources of life's essential building blocks, whether synthesized on early Earth or delivered from the cosmos, we now arrive at a truly pivotal moment in the origin story. The transition from a diffuse, primordial soup of molecules to the first organized, encapsulated structures that could be considered precursors to cells. Life as we know it is cellular. All known living organisms are composed of one or more cells, and each cell is fundamentally defined by its boundary, the cell membrane, which separates its internal environment from the external world. This compartmentalization is essential. It allows the cell to concentrate necessary molecules, maintain specific internal chemical conditions different from the surroundings, protect its delicate machinery, and essentially define itself as a distinct entity. Without such a boundary, the complex molecules of life would simply diffuse away, and coordinated biochemical reactions would be impossible. So how did these first walls arise on the prebiotic Earth? Let's consider level 19, lipid vesicle formation. The primary structural components of modern cell membranes are phospholipids, which are complex molecules. However, simpler lipid-like molecules, such as fatty acids, are known to have been present on the early Earth. Fatty acids have been synthesized in laboratory experiments simulating prebiotic conditions, for example through Fischer-Tropsch type synthesis in hydrothermal systems, and, as we've discussed, they have been found in carbonaceous meteorites, indicating an extraterrestrial source. A fatty acid molecule has a distinctive structure, a long hydrocarbon tail that is hydrophobic, water-fearing, and a carboxyl group head that is hydrophilic, water-loving. Molecules with both hydrophobic and hydrophilic parts are called amphiphiles. When amphiphilic molecules like fatty acids are placed in water, they exhibit a remarkable property. They spontaneously self-assemble into ordered structures to minimize the unfavorable interaction between their hydrophobic tails and the water. Depending on their concentration and shape, they can form micelles, tiny spheres with tails inward, heads outward. Or, more importantly for cell formation, bilayers. A bilayer is a two-molecule thick sheet where two layers of fatty acids align with their hydrophobic tails facing each other, shielded from the water, while their hydrophilic heads face outwards, interacting with the water on both sides. These fatty acid bilayers can spontaneously curve and seal upon themselves to form closed hollow spheres called vesicles. These vesicles are essentially microscopic, self-assembled bubbles or sacs, bounded by a simple membrane. This process requires no biological machinery. It's a consequence of the fundamental hydrophobic effect and the chemical nature of the lipid molecules themselves. The ease with which such vesicles can form from simple, prebiotically plausible molecules is a strong argument for their involvement in the origin of life. They provide a natural and readily available mechanism for compartmentalization. This leads us to level 20, encapsulation of molecules. Once these lipid vesicles begin to form in an aqueous environment that contains other dissolved molecules, like amino acids, nucleotides, or even short strands of RNA or peptides that might have already formed, they can passively trap some of these molecules within their internal aqueous compartment. Imagine these vesicles forming like tiny soap bubbles in a soup. As they seal off, they enclose a sample of whatever was in their immediate vicinity. This encapsulation would have been a random process initially, but it had profound consequences. By trapping molecules inside, the vesicles created a localized, concentrated internal environment. This addresses a major challenge for prebiotic chemistry. In a dilute primordial ocean or pond, the chances of different types of molecules encountering each other frequently enough to react and form more complex systems would be very low. Encapsulation within a vesicle dramatically increases the local concentration of reactants, making chemical reactions and the formation of molecular assemblies much more efficient. It's like moving from trying to have a conversation in a vast, noisy stadium to having it in a small, quiet room. Furthermore, the membrane itself is not just a passive barrier. While it separates the inside from the outside, it can also be selectively permeable, allowing certain small molecules or ions to pass through while retaining larger ones. This could allow the nascent protocell to take in food, simple building blocks, from the environment and expel waste products. The properties of these early membranes would have depended on the specific types of fatty acids or other amphiphiles available. Some studies suggest that membranes made of shorter-chain fatty acids might have been more dynamic and permeable than modern phospholipid membranes, perhaps allowing easier passage of monomers like nucleotides. This brings us to level 21, the dawn of protocells. A vesicle that has encapsulated a collection of molecules, particularly if those molecules include some that can replicate, like self-copying RNA strands, as per the RNA world hypothesis, or catalyze useful reactions like ribozymes, can be considered a protocell or protobiont. This is not yet a true modern cell with all its sophisticated machinery, but it's a crucial intermediate stage, a bounded chemical system capable of some lifelike properties. If a protocell happened to encapsulate a particularly successful set of molecules, for instance, an RNA molecule that could replicate itself and perhaps also catalyze a reaction that helped to stabilize or grow the vesicle membrane or incorporate more fatty acids from the environment, then that protocell would have a selective advantage. It might grow larger. As a vesicle grows, its surface area to volume ratio changes, and it can become physically unstable. Under certain conditions, e.g. gentle shear forces from flowing water, or through the incorporation of more lipids leading to budding, such a growing protocell could spontaneously divide or fragment, forming smaller daughter protocells. If the encapsulated replicating molecules were also distributed to these daughter vesicles, then you have the beginnings of heredity and reproduction at the protocellular level. This is where Darwinian evolution could start to operate on these encapsulated systems. Protocells that were more stable, better at acquiring resources, more efficient at replicating their internal contents, or more successful at dividing, would outcompete others. Over time, this would lead to the selection of increasingly complex and efficient protocellular systems. For example, if an encapsulated RNA molecule mutated in a way that allowed it to also catalyze the synthesis of more fatty acids, that protocell would be better at growing its membrane and reproducing, and its genetic information would spread. Scientists are actively exploring these processes in the laboratory. They can create fatty acid vesicles under simulated early Earth conditions and study how they interact with RNA, peptides, and other molecules. Experiments have shown that RNA can indeed be encapsulated, that vesicles can grow by incorporating more lipids from micelles in the surrounding solution, and that they can be induced to divide by physical forces or changes in the chemical environment. Some research even suggests that RNA itself might be able to influence membrane properties, or that mineral surfaces, like clays, could have played a role in stabilizing early protocells or facilitating the encapsulation process. The transition from a mere collection of organic molecules to these first walled-off protocells represents a monumental leap in organization and individuality. It was the point at which life began to carve out its own distinct niches, its own internal worlds, shielded from the randomness of the external environment. These humble, self-assembled vesicles were the crucibles within which the more complex machinery of life could begin to evolve, the first tentative steps towards the intricate cellular architecture that defines all life on Earth today. The exact sequence and interplay of molecular replication and vesicle formation and division are still areas of intense investigation, but the spontaneous self-assembly of lipids into boundary structures provides a compelling and plausible starting point for the very first homes for life. Having witnessed the potential emergence of the first protocells on a young Earth, our journey now expands its gaze beyond our own planet. If life could arise here, given a specific set of conditions and ingredients, Could it also take root on other worlds orbiting distant stars? This question propels us into the field of astrobiology and the search for habitable exoplanets. To guide this immense search, astronomers and astrobiologists have developed concepts to identify potentially life-friendly environments, starting with Level 22, exploring stellar habitable zones, then considering Level 23, the vital role of liquid water, and finally Level 24, atmospheric needs for surface habitability. At Level 22, exploring stellar habitable zones, we encounter a concept often colloquially referred to as the Goldilocks zone. Formally, the habitable zone, HZ, is defined as the region around a star where an Earth-like planet with a suitable atmosphere could possess liquid water on its surface. This definition is, by necessity, Earth-centric, as our planet is the only place we definitively know life exists, and all known terrestrial life depends fundamentally on liquid water. The idea is that if a planet orbits too close to its star, the intense stellar radiation would cause its surface water to boil away, potentially leading to a runaway greenhouse effect like on Venus, rendering the surface inhospitably hot. Conversely, if a planet orbits too far from its star, it would receive too little energy, and its surface water would freeze solid, making it unavailable for the chemical reactions of life. The habitable zone is that just right orbital band where temperatures could permit liquid water to persist. The location and width of a star's habitable zone are not universal. They depend critically on the star's luminosity, its total energy output, and temperature. Hot, luminous stars, like massive blue or white O, B, and A-type stars, have habitable zones that are much farther out from the star and are significantly wider. However, these stars also have much shorter lifespans, millions to a few 100 million years, which might not provide enough time for complex life to evolve. Though simple life might be possible, cool dim stars, like M-type red dwarfs, the most common type of star in our galaxy, have habitable zones that are very close in and much narrower. While these stars have incredibly long lifespans, trillions of years, offering ample time for evolution, planets orbiting in their close-in HZs face other challenges, such as the risk of being tidally locked, one side always facing the star, leading to extreme temperature differences, and exposure to intense stellar flares, especially when the star is young. Sun-like stars, G-type stars like our Sun, and slightly cooler K-type stars, are often considered prime targets because their habitable zones are at moderate distances and they have lifespans of billions of years, offering a stable environment for life to potentially arise and evolve. Earth, of course, resides comfortably within our Sun's habitable zone. This brings us to Level 23, the vital role of liquid water. Why is liquid water considered so indispensable? Water, H2O, has a unique set of properties that make it an exceptionally good solvent and medium for life as we know it. Excellent solvent. Water is a polar molecule, meaning it has a slight positive charge on its hydrogen atoms and a slight negative charge on its oxygen atom. This polarity allows it to dissolve a vast range of other polar and ionic substances, facilitating the transport of nutrients and waste products within cells and enabling the complex chemical reactions of metabolism. Liquid range. Water remains liquid over a relatively broad temperature range, 0 degrees C to 100 degrees C at Earth's standard atmospheric pressure, a range that is conducive to the complex organic chemistry of life. High heat capacity. Water can absorb a lot of heat without a large change in its own temperature. This property helps to moderate climates on a planetary scale and to stabilize temperatures within organisms. Density anomaly. Unlike most substances, water is less dense as a solid ice than as a liquid. This means ice floats, insulating the liquid water below and preventing bodies of water from freezing solid from the bottom up, which is crucial for aquatic life in colder climates. Chemical reactant. Water participates directly in many essential biochemical reactions, such as hydrolysis, breaking down molecules by adding water. While scientists speculate about the possibility of life based in other solvents, like methane on Titan or ammonia on colder worlds, water's combination of properties makes it the most versatile and widely accepted candidate solvent for life. Thus, The search for habitable exoplanets is, in large part, a search for worlds where liquid water could be stable on the surface. However, simply being in the habitable zone and having the potential for liquid water is not enough. We must also consider level 24, atmospheric needs for surface habitability. A planet's atmosphere plays a critical role in determining its surface temperature and whether liquid water can actually persist. Greenhouse effect. A sufficiently dense atmosphere containing greenhouse gases like carbon dioxide, water vapor, methane, is necessary to trap some of the host star's heat, warming the planet's surface to temperatures where water can be liquid. Without a greenhouse effect, Earth itself would be a frozen ice ball. However, too strong a greenhouse effect can lead to runaway heating, as seen on Venus. Atmospheric pressure. The atmospheric pressure at a planet's surface must be high enough to allow liquid water to be stable. If the pressure is too low, like on Mars today, liquid water will rapidly boil or sublimate into gas, even at temperatures above freezing. Protection from radiation. A substantial atmosphere can help shield a planet's surface from harmful high-energy radiation from its star, like UV and X-rays, and from cosmic rays. An ozone layer, formed from oxygen, is particularly effective at absorbing UV radiation on Earth. Climate regulation. Atmospheric circulation helps to distribute heat around a planet, reducing extreme temperature differences between the equator and poles or between day and night sides, especially important for tidally locked planets. Other planetary factors also contribute significantly to habitability, such as the planet's size and mass, determining its ability to hold an atmosphere and sustain geological activity like plate tectonics, which can regulate climate over long timescales via the carbon-silicate cycle. Its composition, whether it's rocky, and the presence of a global magnetic field to protect the atmosphere from being stripped away by the stellar wind. Think of the habitable zone as the address in a cosmic neighborhood where conditions might be right. But just like knowing a house's address doesn't tell you if it's actually livable inside, being in the HZ doesn't guarantee habitability. You need to know about the house itself, its size, its insulation, atmosphere, its plumbing, potential for liquid water. Scientists are continually refining these concepts. The continuously habitable zone refers to the region where a planet could remain habitable for a significant portion of its star's main sequence lifetime, allowing time for life to evolve. There's also consideration of abiogenesis zones, which might be slightly different from habitable zones, focusing on conditions specifically conducive to the origin of life, which might not be identical to those needed to sustain already established life. The exact implications of all these factors are still being explored, as we only have one definitive example of a life-bearing planet, our own. Our search for just right worlds is an attempt to find other places where the delicate balance of stellar energy, planetary conditions and atmospheric properties might have allowed liquid water to flow and perhaps for life's story to begin anew. Having established the criteria for potentially habitable zones around stars, our cosmic exploration now confronts a monumental challenge. How do we actually find these distant exoplanets, often no larger than Earth, orbiting stars that are light years away? Detecting such small, dim objects next to their overwhelmingly bright parent stars is an extraordinary feat of astronomical observation and ingenuity. The transit method, level 25, has been one of the most successful techniques for discovering exoplanets, responsible for thousands of confirmed detections, largely thanks to missions like NASA's Kepler Space Telescope and the Transiting Exoplanet Survey Satellite, TESS. Imagine you are patiently and precisely monitoring the brightness of a distant star. If a planet orbiting that star happens to pass directly in front of it from our line of sight, an event called a transit, it will block a tiny fraction of the starlight, causing a slight periodic dip in the star's observed brightness. The amount of dimming tells astronomers about the planet's size relative to its star. A larger planet blocks more light, creating a deeper dip. The duration of the transit provides clues about the planet's orbital speed and path. If these dimming events repeat regularly, the interval between them reveals the planet's orbital period, its year. Knowing the orbital period and the star's mass allows astronomers to calculate the planet's orbital distance, which is crucial for determining if it lies within the habitable zone. The transit method is most sensitive to larger planets orbiting close to smaller stars, as they produce a larger, more easily detectable dimming signal. And it requires the planet's orbit to be aligned edge-on to our line of sight, so it only detects a fraction of all existing planets. Next is the radial velocity method, level 26, also known as Doppler spectroscopy, or the wobble method. This technique was responsible for the first confirmed discovery of an exoplanet around a Sun-like star, 51 Pegasi b in 1995, by Michel Mayor and Didier Queloz, who later won the Nobel Prize for this work. As a planet orbits its star, its gravity doesn't just affect the planet also exerts a gravitational tug on the star. Both bodies actually orbit a common center of mass, or barycenter. Because the star is much more massive, this barycenter is usually very close to the star's center, but the star still executes a small wobble in response to its planetary companion. We can detect this wobble by observing the star's spectrum. As the star moves slightly towards Earth in its mini-orbit, its light waves are compressed, shifting its spectral lines towards the blue end of the spectrum, a blue shift. As it moves away, its light waves are stretched, causing a redshift. By precisely measuring these tiny periodic Doppler shifts in the star's spectral lines over time, astronomers can infer the presence of an orbiting planet and estimate its minimum mass, because the inclination of the orbit is usually unknown, and its orbital period. This method is more sensitive to massive planets orbiting close to their stars, as they induce a larger, more rapid wobble. Then we have level 27, direct imaging. This is in many ways the ultimate goal, to actually take a photograph of an exoplanet, to see it as a distinct point of light separate from its star. However, this is incredibly challenging due to the overwhelming glare of the host star, which can be millions to billions of times brighter than the faint light reflected by its planet. It's like trying to spot a tiny firefly sitting right next to a blindingly bright searchlight from many miles away. To achieve direct imaging, astronomers use highly sophisticated techniques. Adaptive optics on large ground-based telescopes actively correct for the blurring effects of Earth's atmosphere in real time, producing much sharper images. Coronagraphs are instruments placed inside the telescope that physically block out the direct light from the star, much like holding your hand up to shield your eyes from the sun, allowing the fainter light from an orbiting planet to be seen. Another concept is a starshade, a large, precisely shaped spacecraft that would fly tens of thousands of kilometers in front of a space telescope to block the starlight before it even enters the telescope. Direct imaging is currently most successful for detecting large Jupiter-like planets that are relatively far from their host stars, reducing the glare problem, and are young enough to still be glowing brightly with the residual heat of their formation, making them more luminous in infrared light. Capturing a direct image of a small, rocky, Earth-like planet in the habitable zone of a Sun-like star remains a major technological aspiration for future, even larger telescopes. Finally, let's touch upon level 28, gravitational microlensing. This technique is based on a prediction of Einstein's theory of general relativity. Gravity bends light. If a star, the lens star, passes almost directly in front of a more distant background star, the source star as seen from Earth, the gravity of the lens star can bend and magnify the light from the source star, causing a temporary characteristic brightening. If the lens star has a planet orbiting it, the planet's own gravity can cause an additional, shorter duration perturbation or blip in this brightening event. Microlensing events are rare, requiring precise alignments, and they are typically one-off events, meaning they usually can't be followed up. However, this method is unique in its sensitivity to low-mass planets, even down to Earth mass or below, at moderate to large orbital distances from their stars, including planets in the habitable zones of M-dwarf stars. It can also detect planets orbiting very distant stars, and even rogue planets that have been ejected from their star systems and are wandering through interstellar space alone. Each of these detection methods has its own strengths, biases, and limitations. The transit method finds planets whose orbits are edge-on. The radial velocity method is biased towards massive, close-in planets. Direct imaging favors large, distant, young planets. Microlensing finds planets far from us and often far from their stars. When multiple methods can be applied to the same planetary system, our understanding becomes much richer. For instance, if a planet is detected by both the transit method, giving its radius, and the radial velocity method, giving its mass, astronomers can calculate its bulk density. This density is a crucial clue to the planet's composition. A high density suggests a rocky terrestrial planet like Earth, while a low density indicates a gas giant like Jupiter or an ice giant like Neptune. The thousands of exoplanets discovered through these ingenious techniques have revealed an astonishing diversity of worlds, far beyond what we see in our own solar system. We've found hot Jupiters orbiting incredibly close to their stars, super-Earths, and mini-Neptunes, planets with sizes and masses between Earth and Neptune for which we have no local analogues, planets orbiting binary stars, and planets in highly eccentric orbits. This diverse planetary census is constantly challenging and refining our theories of how planetary systems form and evolve. And critically, it's providing us with a growing list of potentially habitable worlds to scrutinize more closely in our ongoing search for signs of life beyond Earth. These methods are our remote eyes on the cosmos, patiently sifting through the starlight for the subtle telltale signs of hidden companions. Having explored the ingenious methods astronomers use to find distant exoplanets, let's now bring our cosmic gaze back to our own solar system, specifically to our rusty-hued neighbor, Mars. This planet has long held a special place in human imagination as a potential abode for life. And as we ascend through level 29, sifting through evidence of ancient Martian water, consider level 30, its past atmosphere, and delve into level 31. The search for Martian biosignatures, and level 32, signs of past life, a compelling narrative of a once wetter, possibly more habitable world emerges. At level 29, the evidence for ancient water on Mars is now overwhelming, thanks to decades of exploration by orbiters, landers, and rovers. Spacecraft like NASA's Mars Reconnaissance Orbiter, MRO, have provided stunning high-resolution images, revealing landscapes deeply scarred by fluvial processes. We observe vast dried-up riverbeds, some stretching for hundreds of kilometers. Valley networks that meander and branch like terrestrial river systems, and outflow channels apparently carved by catastrophic floods. Features resembling ancient deltas, where rivers once emptied into larger bodies of water, are clearly visible. Jezero Crater, the landing site of the Perseverance rover, is a prime example, believed to have hosted a lake and a river delta billions of years ago. Curiosity rover, exploring Gale Crater, has found abundant evidence of ancient streambeds and lake deposits, including layered sedimentary rocks that clearly formed in the presence of standing water over extended periods. These are not ambiguous features. They are strong geological fingerprints of a Mars where liquid water once flowed abundantly across its surface. This naturally leads us to level 30, its past atmosphere. For liquid water to have been stable on the Martian surface for extended periods, the planet must have had a significantly thicker and warmer atmosphere than it does today. The current Martian atmosphere is incredibly thin, less than 1% of Earth's atmospheric pressure at sea level, and composed mostly of carbon dioxide. This thin atmosphere provides little greenhouse warming and insufficient pressure to keep water liquid. It would rapidly boil or freeze. However, during the Noachian period, roughly 4.1 to 3.7 billion years ago, when these fluvial features appear to have formed, Mars is thought to have possessed a more substantial atmosphere, likely richer in greenhouse gases like carbon dioxide. This thicker atmosphere could have trapped more solar radiation, raising surface temperatures above the freezing point of water, at least periodically, or in certain regions. What happened to this ancient, thicker atmosphere? The leading theory involves the loss of Mars's global magnetic field. Early in its history, Mars likely had a molten iron core that generated a protective magnetic field, much like Earth's. However, being smaller than Earth, Mars cooled more rapidly, and its internal dynamo eventually ceased, causing the magnetic field to dissipate, probably around 4 billion years ago. Without this magnetic shield, the solar wind, a constant stream of charged particles from the Sun, began to directly impact and erode the Martian atmosphere over billions of years, stripping away lighter gases and gradually thinning it out. This atmospheric loss led to a weakening of the greenhouse effect, causing the planet to cool and dry, transforming it into the cold, arid desert we see today. Given a past with liquid water and a thicker atmosphere, we arrive at the tantalizing prospect of Level 31, the search for Martian biosignatures, and Level 32, signs of past life. If early Mars possessed conditions suitable for the origin and sustenance of life, liquid water, available energy sources, sunlight, geothermal heat, and the necessary chemical building blocks from Martian geology or delivered by meteorites, comets, then it's plausible that life could have arisen there, perhaps contemporaneously with life's emergence on Earth. This hypothetical Martian life would most likely have been microbial. The search for biosignatures on Mars focuses on several key areas. Organic molecules. Rovers like Curiosity and Perseverance are equipped with instruments to detect organic molecules in Martian rocks and sediments. Simple organic molecules, like theophanes, benzene, and methane, have indeed been detected, but their origin is still debated. They could be remnants of ancient life, delivered by meteorites, or formed through non-biological geological processes. The challenge is to find more complex organic molecules or specific patterns that are strongly indicative of a biological origin. Microfossils. The preserved physical remains of ancient microbial cells or their structures would be direct evidence. However, identifying true microfossils is extremely difficult, as non-biological mineral formations can sometimes mimic simple biological shapes, requiring careful analysis. Isotopic ratios. Life on Earth preferentially uses lighter isotopes of certain elements, like carbon-12 over carbon-13. Finding unusual isotopic ratios in Martian carbon-containing compounds could suggest biological processing. Curiosity has found some intriguing carbon isotope ratios in methane and sediments, but more research is needed to confirm a biological link. Mineralogical clues. Certain minerals can be formed or altered by biological activity. Hydrated minerals like clays and sulfates, while primarily indicators of past water, are also good places to look for preserved organic matter or other biosignatures, as they can trap and protect them. Methane fluctuations. Methane in Mars's atmosphere is of great interest because on Earth much of it is produced by living organisms, methanogens. Curiosity has detected puzzling seasonal fluctuations in methane levels and occasional transient plumes. While geological sources like serpentinization can also produce methane, the variability is hard to explain by geology alone, keeping the possibility of a biological source, perhaps deep subsurface microbes, alive, though highly debated. The Perseverance rover is currently playing a key role in this search by collecting and caching samples of Martian rock and soil in Jezero crater. These samples are intended to be returned to Earth by a future joint NASA-ESA Mars sample return mission, likely in the 2030s. Analyzing these carefully selected samples in advanced laboratories on Earth with a full suite of sophisticated instruments will offer an unprecedented opportunity to search for definitive signs of past or even present, if it persists in subsurface refuges, Martian life. Think of Mars as a planetary crime scene investigation. The crime is the disappearance of its ancient habitable environment. The detectives are our rovers and orbiters. They are looking for clues, the dried-up riverbeds, the water-altered minerals, the organic molecules, that tell the story of what Mars was like and whether it ever hosted life. If life did arise on Mars, it may have been driven underground as the surface conditions deteriorated, potentially surviving in subsurface aquifers or brine layers, where liquid water could still exist, shielded from radiation and extreme cold. The search for Martian life, past or present, is not just about finding aliens. It's about understanding whether life is a common cosmic phenomenon or a rare Earth-bound fluke. Finding even fossilized microbes on Mars would profoundly suggest that the universe is indeed predisposed to biology. Our quest for life beyond Earth now takes us further out into the solar system, to the frigid realm of the gas giants, Jupiter and Saturn. Here, orbiting these colossal planets, are a collection of icy moons that have, in recent decades, emerged as some of the most compelling targets in our search for extant life. Far from the sun's direct warmth, these worlds were once thought to be geologically dead and frozen solid. However, spacecraft missions have revealed a startling truth. Several of these moons likely harbor vast oceans of liquid water hidden beneath their thick, icy shells. We begin by exploring Level 33, Europa's potential ocean, then shift to Level 34, Enceladus's plumes. Consider Level 35, Titan's exotic environment, and finally, assess Level 36, their habitability. At Level 33, Jupiter's moon Europa stands out as a prime candidate. Slightly smaller than Earth's moon, Europa's surface is a remarkably smooth, bright shell of water ice, crisscrossed by an intricate network of dark, reddish-brown linear features known as lineae, and regions of chaos terrain, where the icy crust appears to have been broken, rotated, and refrozen. This geologically young surface, indicated by a relative lack of impact craters, suggests ongoing resurfacing processes, hinting at a dynamic interior. The most compelling evidence for a subsurface ocean comes from data gathered by NASA's Galileo spacecraft in the 1990s. Galileo's magnetometer detected an induced magnetic field around Europa, which is best explained by the presence of a global, electrically conductive layer beneath the ice, most likely a salty liquid water ocean. Theoretical models, supported by observations from the Hubble Space Telescope, which has detected tentative evidence of water vapor plumes erupting from Europa, suggest this ocean could be 60 to 150 kilometers, 40 to 100 miles deep, lying beneath an ice shell perhaps 10 to 30 kilometers, 6 to 19 miles thick. If these estimates are correct, Europa's ocean could contain more than twice the amount of water found in all of Earth's oceans combined. The primary energy source keeping this vast ocean liquid is tidal heating, caused by the gravitational interactions between Europa, Jupiter, and the other Galilean moons, Io and Ganymede, which flex and stretch Europa's interior, generating frictional heat. Moving to the Saturnian system, we encounter Level 34, Enceladus's plumes. This relatively small moon of Saturn, only about 500 kilometers or 310 miles in diameter, provided one of the most astonishing discoveries of the Cassini mission. Cassini observed enormous plumes of water ice particles, water vapor, and simple organic molecules erupting from long fissures, dubbed tiger stripes, near Enceladus's south pole. These plumes jet hundreds of kilometers into space, indicating a pressurized subsurface source. Cassini flew directly through these plumes on several occasions, allowing its instruments to analyze their composition. The findings were remarkable. In addition to water and organic compounds like methane and formaldehyde, the plumes contain salts, such as sodium chloride consistent with a salty ocean, and, most tellingly, tiny silica nanoparticles. On Earth, such silica nanoparticles typically form when hot water, above 90 degrees C or 194 degrees, interacts with rock. This is strong evidence for ongoing hydrothermal vent activity on Enceladus's seafloor, where its subsurface ocean is in contact with its rocky core. Tidal heating from Saturn is again the likely engine driving this activity and keeping the ocean liquid. Then there's Level 35, Titan's exotic environment. Saturn's largest moon, Titan, is unique in the solar system for having a dense, nitrogen-rich atmosphere even thicker than Earth's. Its surface, at a frigid 179 degree, 290 degree, features a complex landscape of liquid methane and ethane rivers, lakes, and seas, as revealed by the Cassini-Huygens mission. While liquid water cannot exist on Titan's surface, models strongly suggest that Titan, too, likely possesses a subsurface ocean of liquid water, perhaps mixed with ammonia, which acts as an antifreeze, buried deep beneath its icy crust and its surface hydrocarbon lakes. This ocean is probably kept liquid by a combination of tidal heating from Saturn and radiogenic heat from the decay of radioactive elements within Titan's rocky core. While the primary focus for life on Titan often centers on the potential for exotic, non-water-based life in its surface. Methane lakes, the presence of a subsurface water ocean, adds another layer of potential habitability. This brings us to Level 36, Their habitability. Could these hidden ocean worlds actually harbor life? The key ingredients often cited for life as we know it are liquid water. Europa, Enceladus, and likely Titan all appear to possess this in abundance beneath their icy shells. Essential elements. CHNOPS. The rocky cores of these moons would provide many of the necessary elements, the organic molecules detected in Enceladus's plumes and expected to be present in Europa's ocean, potentially delivered by comets, asteroids, or formed through internal processes, confirm the availability of carbon, hydrogen, nitrogen, and oxygen. Sulfur and phosphorus would also likely be leached from the rocky seafloor. Energy source. While sunlight doesn't penetrate to these deep oceans, chemical energy is available. Tidal heating provides the warmth to keep the oceans liquid. More importantly, if hydrothermal vents are active on the seafloors of Europa and Enceladus, as strongly suggested for Enceladus, they would release reduced chemicals like hydrogen, methane, and sulfides from the moon's interior. These chemicals, reacting with oxidants potentially present in the ocean water, perhaps delivered from the irradiated surface ice on Europa or from other sources, could provide the energy for chemosynthetic life, similar to the ecosystems found around Earth's hydrothermal vents. The conditions within these oceans, dark, cold, though warmer near vents, and under high pressure, would be extreme. But we know that life on Earth thrives in similar extreme environments. If life can arise and persist around terrestrial hydrothermal vents, it's plausible it could do so in these alien oceans. The life forms would likely be microbial, bacteria or archaea analogues, adapted to harness chemical energy. Future missions are planned to investigate these ocean worlds more thoroughly. NASA's Europa Clipper mission, launching in 2024, will perform multiple flybys of Europa to characterize its ice shell, confirm its ocean's properties, and search for active plumes. Concepts for future landers could even attempt to drill through the ice and deploy submersibles. For Enceladus, missions are being proposed to fly through its plumes with more advanced instruments or even land near the tiger stripes. NASA's Dragonfly mission to Titan, a rotorcraft lander, will explore its surface chemistry and assess its broader habitability. Think of these icy moons as cosmic snow globes with a secret liquid heart. The discovery of their subsurface oceans has dramatically expanded our concept of where habitable environments might exist, pushing the boundaries far beyond the traditional sunlit habitable zones around stars. They offer some of the most compelling and relatively accessible targets in our solar system for searching for extant extraterrestrial life, potentially representing oases where life could have arisen and evolved entirely independently of Earth. Our journey has explored potential life-harboring environments within our own solar system, from ancient Mars to the hidden oceans of icy moons. Now we cast our gaze much farther afield, to the exoplanets orbiting distant stars. Since we cannot physically visit these remote worlds with current technology, we must rely on analyzing the faint light that travels across light years to reach our telescopes. A key goal of this remote sensing is to search for biosignatures, chemical fingerprints in a planet's atmosphere that could indicate the presence of life. We'll examine level 37, detecting oxygen, level 38, methane's significance, level 39, the ozone shield clue, and level 40, other atmospheric signs of life. At level 37, detecting oxygen O2 is a prime objective. On Earth, our atmosphere contains about 21% free oxygen, a highly reactive gas. The overwhelming majority of this oxygen is produced by photosynthesis, the process by which plants, algae, and cyanobacteria convert carbon dioxide and water into organic matter and oxygen, using sunlight as an energy source. Without this constant biological replenishment, Earth's atmospheric oxygen would quickly react with surface rocks and other compounds and diminish. While small amounts of oxygen can be produced through non-biological abiotic processes, for example, the photolysis, breakdown by light of water vapor, H2O, by high-energy stellar radiation, with the lighter hydrogen escaping to space and leaving oxygen behind, or the breakdown of carbon dioxide, CO2, sustaining a dense, oxygen-rich atmosphere like Earth's over geological timescales, is widely considered to require a powerful, planet-scale biological source. Therefore, the detection of significant levels of oxygen in an exoplanet's atmosphere, particularly a planet orbiting within its star's habitable zone, would be a very compelling, though not absolutely definitive, biosignature. Context, such as the type of star and the presence of other gases, would be crucial for interpretation. This leads us to level 38, methane's significance, CH4. Methane is another gas of great interest. On Earth, a significant portion of atmospheric methane is biogenic, produced by methanogenic archaea, microbes that thrive in anaerobic, oxygen-poor environments like wetlands, rice paddies, and the digestive systems of ruminant animals. However, methane can also be produced by geological processes, such as serpentinization, the alteration of ultramafic rocks by water, which can release hydrogen that then reacts with carbon dioxide to form methane, and it can be outgassed from planetary interiors or delivered by comets. The real power of methane as a biosignature often comes when considered in conjunction with oxygen. Oxygen and methane are chemically reactive with each other. In an oxygen-rich atmosphere, methane should be oxidized and destroyed relatively quickly. The fact that Earth's atmosphere contains both abundant oxygen and a detectable amount of methane simultaneously suggests that both gases are being continuously produced. This co-presence represents a state of chemical disequilibrium that is difficult to maintain without ongoing biological activity, replenishing one or both gases. Thus, detecting both oxygen and methane in significant quantities in an exoplanet's atmosphere would be a particularly strong indicator of life. Next, at level 39, the ozone shield clue, O3. Ozone is a molecule made of 3 oxygen atoms. In Earth's stratosphere, ozone is formed when ultraviolet, UV radiation from the sun interacts with oxygen molecules, O2, splitting them into individual oxygen atoms, which then recombine with other O2 molecules to form O3. The ozone layer plays a critical role for life on Earth by absorbing most of the harmful solar UV radiation, which can damage DNA and other biological molecules. The presence of a substantial ozone layer in an exoplanet's atmosphere, detectable by its strong absorption of UV light at specific wavelengths, would indirectly imply the presence of a significant amount of oxygen in the lower atmosphere, as oxygen is the precursor for ozone. Therefore, ozone itself is considered a valuable and sometimes easier to detect biosignature, especially from ground-based telescopes where Earth's own atmosphere blocks some of the direct oxygen signals. Beyond these primary targets, we reach level 40, other atmospheric signs of life. Scientists are considering a range of other potential gaseous biosignatures, nitrous oxide, N2O. On Earth, N2O, laughing gas, is produced almost entirely by microbial processes, particularly denitrification by bacteria in soils and oceans. It has few known abiotic sources in significant quantities: methyl chloride, CH3Cl, and methyl bromide, CH3Br. These are produced by various biological processes on Earth, including by some plants and microbes. Dimethyl sulfide, DMS, and dimethyl disulfide, DMDS. These sulfur-containing compounds are produced in large quantities by marine phytoplankton on Earth and play a role in cloud formation. Ammonia, NH3. While present in the early Earth's atmosphere, in a modern oxygen-rich atmosphere, ammonia is quickly oxidized. Its persistent presence in detectable quantities alongside oxygen could be a sign of biological production, though it is also a major component of the atmospheres of gas giants. Phosphine, pH3. Recently, phosphine gained attention due to tentative and still highly debated detections in Venus's atmosphere. On Earth, phosphine is associated with anaerobic microbial ecosystems, though its exact biological production pathways are not fully understood, and abiotic sources under specific conditions are also being explored. The key to identifying a robust biosignature is often not just the presence of a single gas, but rather an atmospheric composition that is in significant chemical disequilibrium, meaning it contains gases that should rapidly react with each other and disappear unless they are being continuously replenished by an active source, likely biological. Another approach is to look for unusual combinations or abundances of gases that cannot be easily explained by known geological or photochemical processes on that particular type of planet, orbiting that particular type of star. Astronomers use techniques like transmission spectroscopy, analyzing starlight passing through a planet's atmosphere during a transit, and emission reflection spectroscopy, analyzing light directly from the planet, to detect these molecular fingerprints. The James Webb Space Telescope, JWST, is a game-changer in this field, with its ability to probe the atmospheres of exoplanets, especially those orbiting M-dwarf stars, with unprecedented sensitivity in the infrared. However, interpreting potential biosignatures requires extreme caution to avoid false positives. For every potential biosignature gas, scientists must meticulously model all known abiotic processes that could produce it under the specific conditions of the exoplanet being observed. For example, oxygen could accumulate abiotically on a planet that has lost most of its water to space, or on a very hot planet with a CO2-rich atmosphere that is being photolyzed. Methane can be produced geologically. The context of the planet, its size, temperature, type of star it orbits, likely geological activity, is crucial. It will likely require multiple lines of evidence, perhaps the detection of several biosignature gases in disequilibrium and ruling out all plausible abiotic explanations, before the scientific community would confidently claim the detection of extraterrestrial life. Think of it as planetary-scale forensic science, sifting through atmospheric evidence for the chemical echoes of biology. While our search for atmospheric biosignatures aims to detect the chemical byproducts of life, whether simple or complex, another intriguing avenue in our quest for cosmic kin involves looking for direct evidence of technologically advanced extraterrestrial civilizations. At level 41, radio signal searches represent the longest-standing and perhaps most well-known approach to SETI. The rationale is that radio waves can travel vast interstellar distances with relatively little absorption or scattering by interstellar gas and dust. Furthermore, human civilization has been using radio waves for communication and radar for over a century, so it's a plausible technology for other civilizations to develop and use. SETI radio telescopes scan the skies, listening for signals that stand out from the natural cosmic radio background. What makes a signal artificial? Narrow bandwidth. Natural astrophysical radio sources, like pulsars, quasars, or star-forming regions, typically emit radiation across a very broad range of frequencies. An artificial signal, however, especially one designed for communication, might be compressed into a very narrow frequency band to maximize its power efficiency and distinguish it from noise. Detecting such a narrow band signal would be highly suggestive of an artificial origin. Pulses or patterns, signals containing deliberate pulses, repeating patterns or complex modulations that encode information, e.g., mathematical sequences like prime numbers or pictorial information, would be strong indicators of intelligence. Drift rate. A signal originating from a transmitter on a planet orbiting a distant star would exhibit a characteristic change in frequency over time, a Doppler drift, due to the planet's orbital motion and rotation. This predictable drift can help distinguish a true extraterrestrial signal from terrestrial interference. Early SETI efforts like Frank Drake's Project Ozma in 1960 focused on specific magic frequencies, such as the 1420 MHz, 21 centimeter emission line of neutral hydrogen, thinking these might be universal beacons. Modern searches, like those conducted by the SETI Institute's Allen Telescope Array, or the Breakthrough Listen initiative using facilities like the Green Bank Telescope and Parkes Observatory, scan much broader ranges of the radio spectrum and employ sophisticated algorithms to analyze vast amounts of data. Moving to level 42, optical SETI, OSETI, we consider the possibility that advanced civilizations might use light, specifically lasers, for interstellar communication. Lasers can produce highly focused, monochromatic, single-wavelength beams of light that can be incredibly bright, potentially outshining the host star in that narrow wavelength band for brief moments. Such powerful, short-duration laser pulses could be an efficient way to send signals across interstellar distances. SETI projects use specialized telescopes equipped with fast photometers or spectrographs to search for these transient, intense flashes of light that cannot be easily explained by natural astrophysical phenomena like stellar flares or cataclysmic variable stars. The advantage of optical communication is the potential for much higher data transmission rates compared to radio waves. Beyond direct communication signals, at level 43. Considering megastructures, we enter the realm of searching for evidence of large-scale astro-engineering projects that a very advanced Kardashev Type 2 or Type 3 civilization might undertake. The most famous hypothetical example is the Dyson sphere, or more plausibly, a Dyson swarm or ring, proposed by physicist Freeman Dyson. He theorized that a civilization with immense energy needs might eventually construct a vast swarm of orbiting solar collectors around its host star to capture a significant portion, or even all, of its radiant energy. Such a structure would largely obscure the star's visible light, but would re-radiate the absorbed energy as waste heat, primarily in the infrared part of the spectrum. Therefore, a potential technosignature could be a star that appears unusually dim in visible light, but exhibits an anomalously large infrared excess. Searches for Dyson sphere candidates have been conducted by analyzing stellar catalogues for such infrared signatures, though no unambiguous detections have been made. Other speculative megastructure ideas include Shkadov thrusters, giant mirrors to propel a star system, or artificial habitats of immense scale. Detecting such artifacts would imply a civilization far more advanced than our own. Finally, at level 44, other technological indicators, we consider a broader range of potential, though often more subtle or speculative, technosignatures. Industrial pollution. Just as human industry releases artificial chemicals, like CFCs, into Earth's atmosphere, an advanced alien civilization might inadvertently pollute its planet's atmosphere with unusual or complex molecules that have no known natural formation pathway. These could potentially be detected through atmospheric spectroscopy of exoplanets. Artificial illumination. The detection of artificial lights on the night side of an exoplanet, similar to our city lights, would be a strong indicator of technology. This is incredibly challenging due to the faintness of such signals, but might be feasible with future extremely large telescopes. Anomalous heat signatures. Concentrated energy use by a civilization could create heat islands, or unusual thermal signatures detectable in the infrared. Non-natural planetary orbits or configurations. Some have speculated that a very advanced civilization might alter the orbits of planets in its system for resource utilization or habitat creation, leading to configurations that are dynamically unusual or difficult to explain by natural formation processes. Interstellar probes or artifacts. The direct detection of an alien probe or artifact within our own solar system, sometimes called artifact SETI, would be definitive proof, though the chances of such an encounter are considered extremely low given the vastness of space. The search for technosignatures faces significant challenges. We have only one example of a technological civilization, our own, to guide our search, and our own technology is rapidly evolving, meaning our assumptions about what alien technologies might look like could be flawed. Furthermore, the question of intent is crucial. Are we looking for deliberate beacons designed to be found, or are we trying to eavesdrop on internal communications or detect unintentional byproducts of technology? The former would be easier to detect, but requires an assumption of altruism or a desire for contact from the other side. Think of this quest like searching an immense dark forest at night. Radio SETI is like listening for a specific, intelligently patterned bird call. Optical SETI is like looking for a brief, unnaturally bright flash of light. Searching for megastructures is like looking for evidence that someone has rearranged entire sections of the forest. Each method requires different tools and makes different assumptions, but all are part of a comprehensive strategy to determine if other technological minds share the cosmos with us. The detection of any credible technosignature would be a discovery of unparalleled significance, instantly reshaping humanity's understanding of its place and future in the universe. Our journey through the levels of unraveling life's mystery has taken us from the primordial soup to the sophisticated search for both biological and technological signatures across the cosmos. We've seen that the universe appears to be filled with stars, many likely hosting planets, and that the basic building blocks of life seem to be common. Yet despite these seemingly favorable odds and our dedicated searches, we are confronted by a profound and echoing silence. The Fermi paradox, as we've touched upon, is the apparent contradiction between the high estimates of the probability of the existence of extraterrestrial civilizations and the utter lack of contact or conclusive evidence for such civilizations. Physicist Enrico Fermi's famous 1950 question, "Where is everybody?" encapsulates this puzzle. Given the age and vastness of the Milky Way galaxy, billions of stars, billions of years for life to potentially evolve and spread, even conservative estimates suggest that at least some civilizations should have reached a stage of technological advancement capable of interstellar communication or travel, and thus should be detectable in some way. Yet the universe, as far as our instruments can tell, appears devoid of such signals. This great silence is not just a lack of evidence. It is evidence of something profound that we don't yet understand about life, intelligence, or civilization in the cosmos. This leads us to level 46, Great Filter. Theories. The concept of a great filter, proposed by economist Robin Hanson, suggests that there is at least one evolutionary step or barrier along the path from simple non-living matter to a spacefaring, galaxy-colonizing civilization that is so improbable or so difficult to overcome that it effectively filters out almost all potential life from reaching that advanced stage. The critical question is, where does this filter lie in relation to humanity? The great filter is behind us. This is the optimistic view for humanity. It implies that one of the steps we have already passed was incredibly rare. Perhaps the origin of life itself, Abiogenesis, is an extraordinarily unlikely event. Or maybe the transition from simple prokaryotic cells to more complex eukaryotic cells, which on Earth took over 1 billion years, is the filter. Other candidates include the development of multicellularity, sexual reproduction, or human-level intelligence and tool use. If the great filter is indeed in our past, it means that humanity is exceptionally rare, perhaps one of the very first, or even the only, technological species to have made it this far in our galaxy. This would explain the silence, as there's simply no one else or very few others out there to make noise. The great filter is ahead of us. This is a far more sobering, even ominous prospect. It suggests that the universe might be teeming with planets where life arises and even evolves some level of intelligence, but that there is a common barrier that prevents these civilizations from surviving long enough to become interstellar, or from avoiding self-destruction. This future filter could be anything from catastrophic wars with advanced weaponry, e.g., nuclear or biological, irreversible environmental collapse due to unsustainable resource exploitation, the unintended consequences of powerful new technologies like runaway artificial intelligence, nanotechnology disasters, or genetically engineered plagues, or even a natural cosmic catastrophe that civilizations are unlikely to survive. If the great filter lies in our future, then the silence of the cosmos could be a grim warning, suggesting that the path we are on is one that few, if any, civilizations successfully navigate for long. We are currently experiencing the great filter. It's also possible that humanity is currently in the process of traversing a great filter, and our future is uncertain. Perhaps the transition to a sustainable, multiplanetary species is the filter. Beyond the great filter, there are numerous other proposed explanations at level 47, reasons for non-detection, which assume that extraterrestrial civilizations might exist, but we simply haven't found them yet, or can't. They are too far away or too different. The galaxy is vast. Even if many civilizations exist, they might be so distant that their signals are too weak to detect, or interstellar travel is simply too impractical for widespread colonization or contact. Their biology, technology, or modes of thought might be so fundamentally alien that we wouldn't recognize their signals or activities even if we encountered them. They might communicate using methods we haven't yet discovered. e.g., neutrinos, gravitational waves, or physics beyond our current understanding. They choose not to communicate or interfere. Advanced civilizations might have reasons for remaining silent or hidden. The zoo hypothesis suggests they might be observing us without interfering, much like we observe animals in a protected nature reserve, waiting for us to reach a certain level of maturity. The prime directive concept from Star Trek posits an ethical principle of non-interference with less developed civilizations. Perhaps they see no benefit in contacting us, or even view emerging civilizations as potentially dangerous or disruptive. They exist for only a short time. The brief window hypothesis. The period during which a civilization is technologically active and producing detectable signals might be relatively short compared to cosmic timescales. They might advance beyond radio, become post-biological, or simply lose interest in outward expansion or communication. If this window of detectability is brief, the chances of two such windows overlapping in time and space are small. We haven't looked long enough or in the right places. Our systematic searches for techno-signatures have been ongoing for only a few decades, a mere blink in cosmic time. We've only surveyed a tiny fraction of the available parameter space: stars, frequencies, signal types, sky coverage. It might simply be a matter of needing more time, more sensitive instruments, and broader search strategies. Economic or resource limitations. Interstellar communication or travel might be prohibitively expensive or resource-intensive, even for advanced civilizations, making such endeavors rare. They are all listening, no one is transmitting. Perhaps everyone is following a cautious SETI strategy, primarily listening rather than actively broadcasting, making the galaxy quiet. Catastrophic events are common, beyond self-destruction. Perhaps natural cosmic events like gamma-ray bursts, nearby supernovae, or asteroid impacts are frequent enough on galactic timescales to regularly sterilize regions or set back the development of advanced life. Think of the Fermi paradox as a cosmic whodunit, or rather a why isn't it happening. The crime is the absence of expected alien activity. The suspects are the various proposed solutions. Each solution carries profound implications for our understanding of life's prevalence, the future of civilization, and our own place in the universe. The Great Silence itself is a powerful data point, telling us that something about our intuitive assumptions regarding the proliferation of advanced life is likely wrong. It urges us to consider that either the emergence of technological intelligence is far rarer than we might like to think, or that the path of such civilizations is far more perilous or alien than we can easily comprehend. The puzzle remains a profound cosmic mystery that continues to fuel both our scientific searches and our deepest philosophical ponderings. As our journey through the levels of unraveling life's mystery approaches its gentle conclusion, and we prepare to drift into sleep, we take a final leap of imagination, venturing into the truly speculative realms of what life could be, beyond the familiar confines of our earthly experience. This leads us to level 48, daring to imagine non-carbon life, then level 49, life in other solvents, and culminates in level 50, reflecting on life's ultimate cosmic mystery and the enduring awe it inspires. At level 48, daring to imagine non-carbon life, we challenge the very foundation of terrestrial biology. All known life on Earth is carbon-based. Carbon is exceptionally versatile. Its atoms can form 4 strong covalent bonds with other atoms, including other carbon atoms, allowing for the creation of an astonishing variety of complex and stable molecules. Long chains, intricate rings, and branched structures that form the backbone of proteins, nucleic acids, carbohydrates, and lipids. But is carbon the only possible basis for complex molecular life? The element most often proposed as an alternative is silicon, Si. Silicon sits directly below carbon in the periodic table, and like carbon, can form 4 bonds. It's also very abundant in rocky planets. Think of silicate minerals, which make up most of Earth's crust. Could silicon-based life exist? While theoretically intriguing, there are significant challenges. Silicon-silicon bonds are generally weaker than carbon-carbon bonds. making long, stable silicon chains, silanes, less robust, especially in the presence of reactive compounds like water or oxygen. When carbon oxidizes, e.g. during respiration, it forms carbon dioxide, CO2, a gas that can be easily exchanged with the environment. When silicon oxidizes, it forms silicon dioxide, SiO2, essentially sand or quartz, a solid. Imagine trying to exhale tiny grains of sand. This makes it difficult to envision a silicon-based metabolism that could easily manage waste products in an aqueous, oxygen-rich environment. However, under very different conditions, perhaps at extremely low temperatures where silanes might be more stable, or in a non-aqueous solvent that doesn't readily break down silicon compounds, some of these limitations might be overcome. Perhaps silicon-based life would have a crystalline or semi-crystalline structure with a much slower metabolism than carbon-based life. Its forms and functions would be utterly alien to us. Other, even more exotic, elemental bases for life have been speculated upon, like boron or sulfur, but they present even greater chemical hurdles. This leads naturally to level 49, life in other solvents. Just as carbon is central to Earth life, liquid water is our universal solvent, facilitating the countless biochemical reactions within our cells. But could life exist in other liquid media, especially on worlds where water itself cannot be liquid? Ammonia, NH3. Like water, ammonia is a polar solvent and can dissolve many organic compounds. It remains liquid at much lower temperatures than water, from 78 degrees Celsius to 33 degrees Celsius at standard pressure, which could make it suitable for life on colder planets or moons. Life in liquid ammonia might use different types of cell membranes and would have a very different biochemistry. Methane, CH4, and ethane, C2H6. These hydrocarbons are abundant as liquids on the surface of Saturn's moon Titan, where temperatures are around 179 degrees Celsius, 290 degrees Celsius. Methane and ethane are non-polar solvents, meaning they would dissolve different kinds of substances than water. Life based in hydrocarbon solvents would likely need cell membranes made of different materials, perhaps based on nitrogen-containing organic molecules called azotosomes, as has been hypothesized, and would have a very different, much slower metabolism due to the extreme cold. Such life might breathe hydrogen and eat acetylene from Titan's atmosphere. Sulfuric acid, H2SO4. While highly corrosive to Earth life, concentrated sulfuric acid can act as a solvent in the cloud decks of Venus, where temperatures and pressures are extreme. Some speculative ideas have considered whether complex chemistry or even a form of life could exist within these acid droplets. If life could indeed be based on different elemental backbones or utilize different solvents, its forms, metabolisms, senses, and even its intelligence, if it evolved, could be so radically different from ours that we might struggle to even recognize it as life. Our search strategies, currently heavily biased towards life as we know it, might completely miss such exotic manifestations. And so we arrive at level 50, reflecting on life's ultimate cosmic mystery and the enduring awe it inspires. Throughout these 50 conceptual levels, from the definition of life to the Fermi paradox and beyond, one thing becomes abundantly clear. The origin and prevalence of life in the universe remain among the most profound and largely unanswered questions facing science and humanity. We still don't know the precise pathway by which life arose from non-living matter on Earth, though we have many plausible hypotheses and encouraging experimental results. We don't know if life is a rare cosmic accident or a common, perhaps even inevitable outcome of planetary evolution when conditions are right. We don't know if any life beyond Earth is simple or complex, microbial or intelligent, familiar or utterly alien. We don't know if the Great Silence is a sign of our uniqueness, a warning of future peril, or simply a reflection of our current technological limitations in listening to a potentially bustling cosmos. This enduring mystery is not a cause for despair, but rather a source of deep wonder and a powerful motivator for continued exploration and research. It fuels our curiosity, drives our technological innovation, and expands our philosophical horizons. The quest to understand life's origin and to search for its presence elsewhere is a fundamentally human endeavor, reflecting our innate desire to know our place in the grand scheme of things. Think of the universe as an immense, ancient library filled with countless volumes. We have so far only managed to read a few pages from a single book, the story of life on Earth. The prospect that other books, with entirely different stories of life, might be waiting on the shelves is what keeps us turning the pages, peering into the darkness, and listening for the faintest whispers from distant worlds. The journey itself through these levels of understanding is as important as any final answer, for it continuously reshapes our perception of ourselves and the cosmos we inhabit. The mystery is vast, the questions profound, and the sense of awe they inspire is a beautiful state in which to drift off to sleep. And now, as our ascent through these 50 levels of cosmic inquiry reaches its gentle summit, allow your mind to drift, to float, to simply be. We have journeyed from the very definition of what it means to be alive, through the fiery crucible of primordial Earth, where the first building blocks simmered, to the tentative assembly of the earliest cells. We ventured forth to explore the just right conditions on distant worlds, the ingenious ways we search for those hidden planets, and the tantalizing whispers we seek from their atmospheres or technologies. We confronted the profound silence of the cosmos and dared to imagine life forms utterly unlike our own. Through all these levels, the enduring thread has been the profound mystery of life itself, its tenacity, its adaptability, its potential ubiquity, and its ultimate origin. The universe, in its incomprehensible vastness and age, holds these secrets close, revealing them only slowly, piece by painstaking piece, to our persistent curiosity. Know that the quest for understanding continues, a quiet, collective human endeavor stretching across generations. Let the complexities we've touched upon dissolve now, like distant starlight blending into the velvet canvas of the night sky. Feel the gentle rhythm of your breathing, a simple, profound anchor in this vast universe. There is a deep peace in acknowledging the immensity of what we do not know, and a quiet joy in the wonder of what we might one day discover. For tonight, your journey is complete. The cosmos will continue its silent, magnificent unfolding, and the mystery of life will await your waking thoughts should you choose to revisit it. Sweet dreams.